Welcome to a deep dive. We're going to be taking a really deep dive into canine cerebral circulation.
Okay.
You're really looking to understand this on like a doctor's level. You want to like really get into the like super intricate details
Yeah.
of the canine cerebral vascular system, especially as it relates to those like fascinating figures and tables from that 2024 review article in Active Veterinary Brana.
Okay.
So let's get to it.
Yeah, absolutely. This review article really is comprehensive. It's becoming, increasingly important to understand the nuances of canine cerebral circulation as intracranial interventions become more common in, vet med. Having a deep understanding is really the foundation for accurate diagnosis and effective treatment.
Yeah. It seems like the article really emphasizes how the canine cerebral vascular system,
Right.
or CVS, isn't just about delivering oxygen and nutrients.
Precisely. Yeah. It's a complex three-dimensional structure with a range of crucial roles. It also plays a critical part in cerebrospinal fluid reabsorption, maintaining stable intracranial pressure, and even regulating brain temperature.
Wow.
The system is broadly divided into arterial and venous parts, interconnected by capillaries, where the real exchange happens.
So starting with the arterial side,
Okay.
what are the key takeaways?
The canine brain receives its blood supply primarily from 5 pairs of arteries.
Okay.
The internal carotid arteries, the vertebral arteries, the rostral cerebellar arteries, the middle cerebral arteries, and the caudal cerebral arteries.
Wow.
4 of these artery pairs come together to form the circle of Willis, a vital structure that provides collateral circulation, ensuring the brain gets blood even if one pathway is blocked.
And I noticed the article highlighted that dogs exhibit posterior circulation dominance.
Yes, that's a key distinction. In dogs, a larger portion of blood flow to the brain comes from the vertebral arteries compared to the carotid arteries.
That's interesting. Are there any other like unique features of the canine cerebral arterial system?
Absolutely. The existence of extra intracranial arterial anastomoses.
Okay.
Essentially, backup connections between arteries outside and inside the skull is quite significant. These anastomoses contribute to the low incidence of ischemic strokes in dogs.
It's fascinating how those like anatomical variations can really impact like clinical outcomes.
Right.
The article also mentioned that the circle of Willis isn't always uniform.
Right. Studies by Tanaka et al.
Okay.
In 2018 and Tanuma back in 1981 have shown quite a bit of variation.
Oh, wow.
For example, Tanuma found a single rostral communicating artery in about 9% of dogs, while a complete circle was observed in only about 55%.
Wow, that's interesting.
Yeah.
It seems like being aware of these variations would be especially important for like surgical planning.
Absolutely.
Now, moving on to those capillaries we mentioned, the article emphasized their density and like importance for nutrient exchange.
Yes. The canine cerebral capillary network is incredibly dense. It forms this intricate three-dimensional web with a huge surface area for the exchange of nutrients, oxygen, and waste products between blood and brain tissue.
The article went into detail about the unique structure of these capillaries,
Right.
highlighting their non-fenestrated nature and thin walls.
That unique structure is critical because it forms the foundation for the blood-brain barrier, or BBB. Essentially, it's a highly selective barrier that carefully regulates what can enter and exit the brain.
Yeah.
Tight junctions between the endothelial cells of the capillaries, along with parasites and specialized transport mechanisms, ensure that only specific molecules can cross.
So it's like a gatekeeper.
Yeah.
For the brain.
Yeah.
Keeping it safe from potentially harmful substances. What happens to the blood after it passes through the capillaries? How does it exit the brain?
That's where the venous system comes in. Once the blood has delivered its oxygen and nutrients and picked up waste products, it's collected by small veins called venules. These venules merge into larger veins, eventually leading to bridging veins that empty into the dural venous sinuses, or DVSs.
The article mentioned that these cerebral veins are quite different from arteries.
Yes. Cerebral veins have thinner walls, and importantly, they lack valves. Unlike arteries, which have a strong muscular layer and valves to prevent backflow, veins rely on pressure gradients and other mechanisms to ensure blood flows in the right direction.
And the DVSs themselves seem quite intricate.
Yeah.
Based on like figures 2 and 3 in the article.
They are. The DVSs are organized into dorsal and ventral groups.
Okay.
Each containing specific sinuses, strategically positioned to drain blood from different regions of the brain. The dorsal group, which you can see clearly in Figure 2, includes the prominent dorsal sagittal sinus running along the midline, the straight sinus, and the paired transverse sinuses.
And Figure 3 shows the ventral group, the cavernous sinuses, the sigmoid sinuses, the basilar sinus, and others, forming a network beneath the brain.
Yeah.
It's incredible how everything is so interconnected.
It really is. And just like the arterial system, there can be variations in the venous system, influenced by factors like breed and skull shape.
For example, the article mentioned that the dorsal sagittal sinus can have different shapes in brachycephalic dogs with their flat faces compared to dolichocephalic dogs, known for their long noses.
Exactly. And you'll find variations in other sinuses as well, like the straight sinuses, the intercavernous sinuses, and the intrabasilar sinuses. But ultimately, despite these variations, the venous system as a whole performs the crucial task of directing deoxygenated blood from the brain back into the systemic circulation.
So we've covered the arteries and the capillaries, and now the veins. It's amazing how these different components work together to ensure efficient blood flow to and from the brain. What about the actual dynamics of blood flow? How is that regulated in dogs?
That's a crucial aspect. The brain, as you know, has a very high metabolic demand and requires a constant, well-regulated blood supply to function properly.
The article provided some typical hemodynamic parameters for dogs, such as heart rate, blood pressure, and the percentage of cardiac output received by the brain. It's remarkable how much blood the brain actually needs.
It is. And it's not just about the amount, it's also about maintaining the right pressure and flow. The brain relies on sophisticated mechanisms to ensure a consistent and stable blood supply, including autoregulation, where blood vessels adjust their diameter in response to changes in blood pressure.
And the article mentioned how factors like carbon dioxide levels, pH, and oxygen levels can also influence blood flow. It's like a complex feedback system, constantly fine-tuning things to keep the brain optimal condition.
That's a great way to put it. Now, all of this, the blood volume, pressure, and flow, plays a significant role in regulating intracranial pressure, which is another crucial aspect of canine cerebral circulation.
And that's where the fixed volume of the canine skull comes into play, as the article highlighted. With the skull being like a rigid container, any change in the volume of one of the elements inside, like blood, brain tissue, or cerebrospinal fluid, affects the others.
Exactly. The article delved into the Monro-Kelly doctrine, which describes this crucial relationship. Any increase in one element must be compensated for by a decrease in another to prevent a dangerous rise in intracranial pressure.
So, for instance, if there's a bleed within the brain, that increased blood volume could put pressure on brain tissue or displace cerebrospinal fluid.
Precisely. Maintaining that delicate equilibrium within the skull is absolutely essential for brain health. The article also went into some detail about how factors like body position and pressure changes in the chest or abdomen can influence intracranial pressure. It's a complex interplay of forces, and that complexity extends to the mechanisms that maintain blood flow in the intracranial venous system, a particularly fascinating aspect covered in detail within the article.
It did seem like a lot was going on there. To keep that blood moving, what stood out to you as the key points?
Well, you have the basic pressure gradients between the venules and the larger veins, along with the pulsations of the brain itself, which help to propel blood forward. Even the dog's body position can have an influence on venous return. But one of the most interesting mechanisms is the Starling resistor effect.
I remember that being mentioned. How does that work exactly?
It's quite clever, really. Imagine a collapsible tube, like a vein, passing through a compartment with a fixed pressure. That's essentially what's happening with these bridging veins. As they pass through the dural sinuses, the Starling resistor effect ensures that the pressure within the veins remains higher than the pressure in the surrounding sinuses, preventing them from collapsing and ensuring continuous blood flow.
So it's like a built-in safety mechanism to prevent blood from getting backed up in the brain.
Precisely. And it plays a critical role in regulating cerebrospinal fluid resorption as well, helping to maintain that crucial balance of fluids within the skull.
This deep dive has really highlighted how interconnected everything is within the canine cerebral vascular system. The article touched on the clinical significance of venous drainage, particularly the risk of cancer cells or emboli spreading through this network.
Yes, that's an important point, because the craniospinal venous system is essentially valveless. There's potential for things to move in both directions.
Oh, wow.
A tumor that originates in the abdomen, for example,
Okay.
could potentially spread to the brain through the venous system, or a blood clot could travel from the legs to the brain, leading to a stroke.
That's a sobering reminder
Yeah.
of how vulnerable this system can be. The article also mentioned using ultra-fast computed tomography to measure regional cerebral blood flow in dogs.
That's a powerful tool for understanding how blood flow varies in different areas of the brain and how it's affected by different conditions. It allows us to visualize blood flow in real time, giving us valuable insights into brain function.
The numbers the article gave on, like, cerebral blood volume distribution
Right.
were interesting. Around 15% in the arteries, 40% in the veins and sinuses, and 45% in the microcirculation.
Those percentages underscore the importance of healthy microcirculation, which is essentially the flow of blood through the smallest vessels, the capillaries and venules. That's where the critical exchange of oxygen, nutrients, and waste products occurs. So maintaining good blood flow through these tiny vessels is crucial for optimal brain function.
And we can't forget the role of the autonomic nervous system in regulating cerebral blood flow. As the article highlighted, it's fascinating how the sympathetic and parasympathetic branches work together, with sympathetic stimulation generally causing vasoconstriction and parasympathetic stimulation leading to vasodilation.
It's like a delicate dance between these two branches, constantly adjusting blood flow to meet the brain's ever-changing needs. And what's interesting is that this regulation isn't uniform across the entire brain.
Okay.
Different regions have different levels of sensitivity to autonomic control, allowing for fine-tuning of blood flow based on the specific metabolic demands of each area.
It's incredible how the brain can, like, orchestrate such precise control.
Yeah.
The article even mentioned how certain neurotransmitters, like norepinephrine and acetylcholine, can directly influence the tone of cerebral blood vessels. It really highlights, like, the interconnectedness of everything.
It does, and that interconnectedness extends beyond the brain itself. Systemic factors like overall blood pressure and cardiac output play a crucial role in cerebral circulation.
Right.
The brain doesn't exist in isolation. It's part of a larger integrated physiological system.
The article also mentioned the concept of cerebral vascular resistance,
Okay.
essentially the opposition to blood flow within the brain.
Right. It's analogous to resistance in an electrical circuit. The diameter of the blood vessels, the viscosity of the blood itself, and even intracranial pressure can all influence vascular resistance. Any alteration in these factors can significantly impact blood flow to the brain.
That makes sense. A narrower vessel, or like thicker blood, would naturally increase resistance. The article used like a thermostat analogy to describe cerebral autoregulation, which I thought was helpful.
Yes, that's a good comparison. Cerebral autoregulation is the brain's way of maintaining a consistent blood supply, even when systemic blood pressure fluctuates.
Right.
Much like a thermostat maintains a steady temperature despite changes in the surrounding environment.
So if blood pressure goes up, cerebral blood vessels constrict to reduce flow.
Right.
If it goes down, they dilate to increase flow. It's like a self-adjusting system. Keeps things in balance.
Exactly. And this mechanism is vital for protecting the brain from damage due to either too much or too little blood flow. However, it's important to remember that even autoregulation has its limits. If blood pressure fluctuations are too extreme or prolonged, this mechanism can fail, leading to potentially serious consequences.
The article also emphasized the importance of cerebral perfusion pressure, which is essentially the pressure gradient driving blood flow to the brain.
Yes. It's the difference between the mean arterial pressure and the intracranial pressure. If this pressure gradient is too low, blood flow to the brain can become inadequate, leading to ischemia. Conversely, if it's too high, it can damage delicate brain tissue. Maintaining the right balance is critical.
And of course, as we've discussed, intracranial pressure itself plays a significant role in cerebral blood flow.
Absolutely. Any increase in intracranial pressure, whether due to a tumor, bleeding, or swelling, can compress those cerebral blood vessels and reduce blood flow, potentially leading to a cascade of negative consequences.
And on the flip side, any decrease in cerebral blood flow can deprive the brain of oxygen and nutrients,
Yeah.
potentially leading to ischemia or even cell death. It's a delicate balancing act
Right.
that the body strives to maintain.
It is. The body has a remarkable ability to compensate for changes in intracranial pressure to try and protect the brain. One way it does this is through cerebrospinal fluid regulation.
Okay.
The brain can adjust the production, absorption, and flow of CSF
Okay.
to help stabilize the intracranial environment.
So it's like a pressure valve,
Yeah,
releasing or absorbing CSF as needed
right.
to maintain equilibrium.
Exactly. And another compensatory mechanism is the displacement of venous blood.
Okay.
When intracranial pressure rises, the body can actually squeeze some venous blood out of the skull
Wow.
to make more room for the expanding brain tissue, acting like a temporary pressure relief valve.
It's incredible how the body has these like built-in mechanisms to protect the brain.
Right.
The article also mentioned that the brain can actually adjust its own like metabolic rate to adapt to changes in blood flow.
That's right. When blood flow is reduced, the brain can slow down its activity to conserve energy, a testament to its incredible plasticity and adaptability.
This deep dive has been truly enlightening. We've learned so much from just this one review article about the intricacies of canine cerebral circulation.
We've only just begun to scratch the surface of this complex and fascinating field. There's still so much we don't fully understand about the canine brain.
It's incredible to think about all the like potential discoveries yet to be made.
Yeah.
But this deep dive has definitely provided like a solid foundation for understanding the complexities of this vital system.
It really is remarkable how much we've like uncovered about the canine cerebral vascular system just by like exploring this one review article.
This knowledge is like so crucial for veterinarians when it comes to like diagnosing and treating those like complex neurological conditions in dogs.
Absolutely. Understanding the intricate workings of cerebral circulation, from the tiniest capillaries to the larger veins and sinuses, is essential for making informed decisions about patient care. It lays the groundwork for effective diagnosis, treatment planning, and ultimately better outcomes for our canine companions.
You're absolutely right. Understanding these mechanisms is truly a game changer for like veterinary neurology. And that's the crux of the matter, isn't it? The more we like delve into the complexities of the canine brain, the better equipped we become to safeguard their health and well-being.
It is. And as we continue to learn more, we might even discover new insights that could have implications for human health as well.
Oh, wow.
The parallels between canine and human neurological conditions are striking, and research in one area can often inform the other.
That's a fascinating point. So given all that we've learned about the unique features of the canine cerebral venous system, what research avenues do you think hold the most promise for improving our understanding of neurological diseases and enhancing treatment options?
That's the burning question. There are many directions we could go. I think further investigation into the role of the Starling resistor in different neurological conditions could be very valuable. Does its function change in cases of head trauma or hydrocephalus? How does it respond to different medications?
That's a great point. And I imagine like further exploring those variations in venous drainage patterns across breeds could also yield like valuable insights. Could certain breeds be predisposed to specific neurological conditions based on their unique venous anatomy?
Exactly. And with advancements in imaging technology, we can study these patterns in unprecedented detail, potentially leading to earlier diagnosis and more targeted treatment strategies.
It's exciting to think about the possibilities. We've really journeyed through the intricate landscape of canine cerebral circulation in this deep dive. It's been a privilege to explore this topic with you.
The pleasure has been all mine.
From Vet Neurojar, keep those minds inspired, hearts light, and tails wagging.